रेगुलेशन मैकेनिज्म है, उसकी वो नोडल ऑफिसर है फोर्थ लेवल में, आपको रखा था, मोहन सिंह को रखा है, तो उनकी मीटिंग है अभी। हाँ आगे उसने भेजे होगे ना, सौरभ ने भेजे, में कर रहे हैं आप मत खोलो, पर आपको दे देंगे। लग रही ना यार, पंखा उस दिन भी नहीं चल रहे थे ना यहाँ पे।
Hello everyone. Good morning to all the faculties that have joined this 21 days hands-on training on remote sensing and GIS using QGIS. This training is being conducted under the NAHAB project in the direction of Dr. R.K. Nirmal sir, who is the principal investigator of this project. And this training is being coordinated by Dr. S.K. Sharma sir. So I am requesting Dr. Sharma sir to please brief the participants about this training. Sharma sir, please. Sir.
हेलो, हाँ सौरभ, आवाज आ रही मेरी?
हाँ सर, क्लियर है।
आवाज क्लियर है?
Yes, sir. Clear is. आ रही है सर, आवाज आ रही है।
हेलो, चलिए। हाँ, सभी पार्टिसिपेंट्स को गुड मॉर्निंग। आज से हम ये 21 डेज ट्रेनिंग प्रोग्राम जो कि बेसिक्स ऑफ रिमोट सेंसिंग एंड जीआईएस के ऊपर रखा है, और हमारे ये एग्रीकल्चर यूनिवर्सिटी के जो फैकल्टीज हैं, हमारी यंग फैकल्टीज, और जो जीआईएस में इंटरेस्ट ले रहे हैं, काफ़ी बहुत अच्छा लगा कि जो पार्टिसिपेशन है काफ़ी अच्छा है। इस ट्रेनिंग प्रोग्राम को हम लोगों ने जो डिवाइड किया है वो हैंड्स ऑन ही रखा है। तो आपको ओनली 2 दिन हम जो रखेंगे वो लेक्चर रहेंगे, और उसमें 2 लेक्चर हम यहाँ फैकल्टी से करेंगे और 2 लेक्चर हम बाहर से नेशनल इंस्टीट्यूट ऑफ हाइड्रोलॉजी या आईआईटी से रखेंगे। और कुछ वीडियोस आपको हम कुछ सेशंस में दिखाएंगे एप्लीकेशन पार्ट के, मगर हमारा इसमें मुख्य उद्देश्य जो है इस ट्रेनिंग प्रोग्राम में कि उस चीज को सीखें, और सीखने के लिए बेसिक जो चीज है रिमोट सेंसिंग की, वो छोटी छोटी चीजें हैं, वो पूरी चीज के बारे में इसमें आपको 21 दिन के इस प्रोग्राम में आपको सिखाया जाएगा। तो पार्टिसिपेंट से मेरी एक रिक्वेस्ट ये है कि सभी लोग इसको अटेंड करें पूरे टाइम तक। और दूसरी चीज कि जो आखिरी में एक हमने 17 तारीख को पार्टिसिपेंट के प्रेजेंटेशन का पार्ट रखा है, वो प्रेजेंटेशन का पार्ट जो पार्टिसिपेंट उसमें कहेंगे, अपना प्रेजेंटेशन करेंगे, ट्रेनिंग से सीखते हुए जो चीजें उनको बताया जाएगा, डिफरेंट बातें हैं, तो वो प्रेजेंटेशन वो करेंगे। तभी हम लोगों ने डिसाइड किया है, बिकॉज इट इज 21 डेज ट्रेनिंग प्रोग्राम, जो कि एक सर्टिफिकेट देना है हमें, उसकी कुछ सर्टेन गाइडलाइंस उसकी चीज की, तभी वो वैलिड है, अदरवाइज वो वैलिड नहीं होगा। तो पार्टिसिपेंट बहुत ध्यान दें कि 17-18 तारीख को उनको एक प्रेजेंटेशन इस चीज का देना है, तो वो ठीक से उस चीज को सीखें। और उसके बाद जो पार्टिसिपेंट जो भी कोई व्यक्ति नहीं प्रेजेंट करेगा, हम सर्टिफिकेट उसे ही ले यहाँ से। ये हमारा मतलब हमारी गाइडलाइन भी है, एनएचपी गाइडलाइन में ही है इस तरीके से। ये तो हम बाउंडेड हैं उस चीज के लिए। तो आप कृपया इस चीज को बहुत अच्छे से ध्यान, 75% और एक रिक्वायरमेंट है कि वो 75% आप रिक्वायरमेंट अटेंडेंस की होना चाहिए कि आप और आफ्टरनून सेशन में आपका 2.5 घंटे का
सर आपका वॉइस डिस्कनेक्ट हो।
हेलो।
आप इससे कर लो।
हाँ, मैं आपका वो माइक ठीक पड़ेगा। जी, तो इस हम आपको आफ्टरनून सेशन में प्रैक्टिस सेशन हमारा रहेगा, मॉर्निंग सेशन में हमारा आपको चीजों को करके दिखाएंगे, साथ में आप करते जाइएगा उसको। और प्लीज आप अपना डेस्कटॉप और लैपटॉप, अभी 2 दिन तो आपका थ्योरेटिकल चीजें चलेंगी, मगर आप थर्ड डे हम आपको जो ये जो चीजें हैं ये हमारी यहाँ से आपकी ऑनलाइन हैंड्स ऑन स्टार्ट हो जाएगी। तो आप प्लीज उनको रेडी रखें, सो दैट वो चीजें आप जो हम सिखाना चाहते हैं वो आप पूरी चीजों को सीख सकें और आप उस पर वर्क कर सकें और प्रैक्टिस कर सकें, या प्रॉब्लम आपकी आ रही उन चीजों पर कर सकें। थैंक यू।
Now I'm requesting our PI sir, Dr. R.K. Nema sir, to please address the participants.
Well, Saurav, next program is the introduction of the participants. Saurav?
Yes, sir. Yes, sir. Yes, sir.
Well, you just request all the participants to introduce themselves one by one. You just name them. They can just unmute and tell about this only. What is their name, where from they are, and their discipline and year of experience.
Okay, sir. So please, all the participants, I'm just requesting you to please unmute yourself one by one, and please give your introduction with the name, department, and the number of year of experience you have. So please, you can start it from one by one.
Hello. Hello. Hello, sir. Myself, Dr. M.K. Naik, Mahendra Kumar Naik, Assistant Professor, College of Agriculture, Tikamgarh.
Hello, myself, Somnath Sarvodaya, Assistant Professor, Agroforestry, College of Agriculture, Balaghat. I have experience of 6 years.
Good morning to everybody. Myself, Dr. Lalit Mohan Bal, Assistant Professor at College of Agriculture, Tikamgarh, in Post-Harvest and Food Engineering. I have experience of around 9 years at this college. Thank you very much.
Sir, myself, Dr. Pradeep Misra, Assistant Professor Statistics, College of Agriculture, Pawarkheda. I have experience of 4 years. Thank you.
Good morning, sir. Myself, Dr. Rajmohan Sharma from College of Agriculture, Kanjurmarg, Assistant Professor in Department of Plant Breeding and Genetics, and I have more than 12 years experience as Assistant Professor. Thank you, sir.
Good morning, all of you, sir.
Myself, Dr. Ashish
Kumar Panse,
Assistant Professor, Plant Pathology, College of Agriculture, Ganjvasola.
Good morning, all of you, sir. Myself, Raju Kumar Panse from College of Agriculture, Balaghat, working as an assistant professor, and I have experience 8 years. Thank you.
Good morning to all of you. I am Dr. Sailendra Bhalave, Department of Agroforestry from College of Agriculture, Balaghat, and I have experience 6 years.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Good morning, sir. Myself Rajendra Prasad Dongre, Assistant Professor in Department of Forestry, JNKVV Jabalpur, since from 2007. Thank you, sir.
Good morning,
Good
sir.
morning to all of you.
Good morning, sir.
Myself, Dr. Dhananjay Kathal, Assistant Professor, Department of Plant Pathology, College of Agriculture, Pawarkheda. I have experience in 6 years.
Good morning, sir. Stephen Aberia, Assistant Professor, Department of Extension Education, College of Agriculture, Jabalpur, and I am having experience 30 years.
Good morning, sir. Myself, Dr. Atul Kumar Srivastava, Department of Agronomy. I am from College of Agriculture, Balaghat. I have 7 years experience as an assistant professor. Thank you, sir.
Good morning to all of you. I am Dr. Alpana Singh. I am working as a professor, Department of Food Science and Technology, College of Agriculture, Jabalpur. And I am engaged in teaching, research and extension since last 33 years.
Hello, good morning. Myself, Dr. KGIS, Professor of Plant Pathology,
Myself,
College
Dr.
of
Ashish
Riva.
Kumar,
I have experience of
working as
23
scientist
years.
and pathology.
Hello, good morning, sir. I am Dr. Vivek Bade, Assistant Professor Computer Science at College of Agriculture, Balaghat. I am since here from 2017. Thank you.
Good morning, everyone. This is Sheila Raghuvanshi, Assistant Professor in the Department of Extension Education, College of Agriculture, Tikamgarh. And I'm having the experience in teaching and research less from the 9 years. Thank you.
Good morning, sir. I am Dr. Uttam Kumar Bisen, College of Agriculture, Balaghat, Department Plant Breeding and Genetics. I have experienced 14 years.
Good morning, sir. Myself, Dr. Rishikesh Thakur, Assistant Professor Soil Science from College of Agriculture, Balaghat, and I have experience 6 years.
Good morning, sir. Myself, Dr. Ghanshyam Deshmukh, Associate Professor, College of Agriculture, Department of Agriculture Engineering. I have 13 years experience.
Good morning, sir. Myself, Dr. Amita Sharma. Myself, Dr. Dharna Bisen from Agriculture College, Balaghat, Department of Entomology, and I have experience of 9 years. Thank you, sir.
Good morning, everyone. Myself, Dr. Amita Sharma. I'm working as Assistant Professor in College of Agriculture, Balaghat. I'm from Plant Breeding and Genetics Department, and I have an experience of 6 years in teaching and research. Thank you.
Good morning to all of you. I am Yog Ranjan, Assistant Professor at College of Agriculture, Tikamgarh, presently attached at Ministry of Agriculture and Farmer Welfare, and having an experience of 14 years.
Good morning, everyone. This is Dr. Anil Mishra, working with College of Agriculture, Tikamgarh, and having experience of 13 years in teaching. I am working with Business Studies Department. Thank you.
Good morning, everyone. I am Dr. Surendra Rai, Assistant Professor at College of Agriculture, Balaghat. I have experience of 6 years. Thank you.
Good morning, everybody.
Everyone. My name is Dr. Sharad Bisen. I am from College of Agriculture, Balaghat, Department Horticulture. I having experience of 8 years. Thank you.
Good morning, everybody. I am Dr. Vikram Singh Gaur. I'm at College of Agriculture, Balaghat, Assistant Professor in Agriculture Biotechnology. Next is having experience of 6 years. Thank you.
Thank you, all of you, for introducing yourself.
Hello, morning. Myself, Dr. Rajan Singh Thakur, Department of Food Science and Technology, College of Agriculture, Jawaharlal Nehru Krishi Vidyalaya, Jabalpur.
Hello. Morning, everyone.
Good
Myself,
morning.
Dr. Ashish Kumar, scientist at Seed Technology Research Center, College of Agriculture, Jabalpur.
Hello. Good morning to all of you. Myself, Dr. Naresh Kumar Visen, Associate Professor, Plant Breeding and Genetics, College of Agriculture, Balaghat. I am having 20-year experience.
I hope everyone has introduced himself. Now I'm requesting our PI sir, Dr. R.K. Nema sir, to please brief the participants, please. Dr. Nema sir.
Hello, good morning.
Thank you.
Saurabh, respected Dean sir. Am I audible?
Yes, sir, you are audible, sir.
Respected Dean sir, OPI Dr. M.K. Awasthi. Dear participants, Tikamgarh, Balaghat, Jabalpur, say all the participants जो well educated हैं और well experienced हैं, and therefore they have been brought under the umbrella of National Higher Agricultural Education Project and for and remote sensing in GIS application, basics of remote sensing in GIS application. Different fields. What are the capabilities you can do from your own? What are the softwares available? What are the data sources available? And the trainers will orient you towards these data sources, towards these open sky sources, and you can hands-on. You can practice those sessions in the afternoon. What Dr. Sharma told that this training should not become a formality. It should be a real training, and then only we will achieve our objective. In the forenoon session, the demonstration as well as the lecture will be conducted. As well as you can practice little Yeah, with this brief saying, I welcome again you all. I extend my thanks to Dean Dr. Atul Srivastava, OPA Dr. Avasti, and coordinator of this training, Dr. S.K. Sharma.
Voice is not clear, sir. We are unable to listen anything. Please.
आवाज बंद हो गई थी क्या? आवाज बंद थी?
सर बहुत डिस्टरबेंस है सर, वेबएक्स पे बहुत ज्यादा।
अच्छा, अभी आवाज बंद हो गई थी क्या?
बार सर बहुत कम आवाज आती है। तो बेटर होगा जूम में कंडक्ट किया जाए।
अगर ये संभव नहीं होगा तो गूगल मीट पे ट्रांसफर करेंगे इसको बाद, ठीक है?
सर इको भी हो रहा है वॉइस आपका।
हेलो
सर ये वॉइस इको भी हो रहा है और क्लियर भी नहीं आ रहा सर, बिल्कुल भी समझ में नहीं आ रहा है।
आप अपने माइक बंद करें, आप अपने सब माइक बंद करें, सारे माइक बंद करें, वीडियो ऑन रखें। आपके सब माइक ऑन है इसलिए इको हो रहा है। So again I am requesting you all, अभी मेरी आवाज क्लियरली आ रही है? एक कोई बताए? Okay, so I am again and again requesting, please be attentive in all the days, all the time, all the session. We have kept it as a rule that at least 75% attendance should be there as in our academic works, but I want that you must be 100% here and you must learn, because this is, we are learning for ourselves, for the university, for the college, for our profession. So be attentive, and then when you complete this training, it will form a platform for further training at different institutes in the national cadre or at the international level. So we have the provision in the project to send the prominent candidates outside also. So please be attentive, and whatever the team is saying in the forenoon session, you have to practice in the afternoon session. So please cooperate and learn many things. Thank you. And now I request our Dean, Dr. Atul Srivastava, to say a few words on this occasion, as he is has been kind enough for being present at this occasion. Dr. Atul Srivastava.
Good morning, everybody. Thank you, Dr. Nema, sir. Good morning, all participants, those are attending this training of our college, university. This is a capacity building for the hands-on training for the remote sensing and GIS. In this both field is very much important since last decades. But this is an opportunity to all of us to learn these things, and I learned from these professors like Professor Nema, Avasti, and Dr. Sharma. And you people are very fortunate that you have very good professor here. And for here, this is not for you only, it is for our, not only for our university only, this is for whole nation only. We want to enhance the technology in our nation. So if you go through in this training for its application, uses, how to analyze and analyze the things according to our requirements, It's helped a lot for all of us, and it is good for the research as well as for the students also. So my best wishes to all participants, and my request to all my university colleagues and participants, they may sincerely attend this training. You have good opportunity to learn by the learned people here. Those are famed in not nationally but internationally also, by their publication, by their trainings and their experience. So all the best of wishes to all, best of luck, and thank you very much.
Thank you very much, sir, for your kind words. Now I'm requesting Dr. R. Shivaram Krishnan to please start the presentation, sir.
Well, Saurabh, before just requesting Dr. Shivarama Krishnan, let me just say thanks to the DIAS who has come here for this training.
Yes, sir. Request you, sir, please do the vote of thanks to all speakers.
देखिए, ये जो ट्रेनिंग का मुख्य पर्पज ये हुआ है कि हमारे स्टूडेंट्स जो एमटेक, एमएससी, पीएचडी किसी भी डिसिप्लिन से हों, उनके जो स्टूडेंट प्रोजेक्ट्स हैं, उन प्रोजेक्ट्स में रिमोट सेंसिंग और स्पेशल डेटा का जो भी यूज है वो इंट्रोड्यूस किया जाए. सारे ऑब्जेक्टिव उनके ना हो, कम से कम एक ऑब्जेक्टिव हो. दे शुड include at least one objective or any kind of the use in remote sensing. So for this, there has been a training, there has been a meeting arranged under the PME-cell. PME-cell is headed by the Dean Faculty of Agriculture, and whose members are DS, DI, DRS, everybody was there. So it was decided to give the preliminary knowledge to all the interested teachers who are willing to have their students with the knowledge of spatial data use. So this particular training, देखिए ये बाकी ट्रेनिंग आपकी रिमोट सेंसिंग की आपकी बहुत सारी आगे सेकंड स्टेप में होंगी, पर ऐसा देखा गया और समझा गया कि हमारे पास बेसिक नॉलेज की कमी है, तो उसको करने के लिए ये सेंटर ही ने अपने आप को आगे बढ़ाया कि ओके, वी आर प्रोवाइडिंग हैंड्स ऑन ट्रेनिंग, वेरी थॉरोली वी आर गिविंग यू, बहुत स्लो आराम से एक एक चीज बताते हुए, जिससे कि कम से कम आपको जो बेसिक नॉलेज है वो हो जाए. रिमोट सेंसिंग में सबसे बड़ी हिच होती है, किसी भी ट्रेनिंग में जाते हैं तो आपसे बोला जाता है कि आप रिमोट सेंसिंग की बेसिक नॉलेज की बात बोलिए, तब हम आपको इंट्रोडक्शन आगे बताएं, तो उसमें बड़ी प्रॉब्लम जाती है. इसीलिए ये बेसिक ट्रेनिंग यदि आप लेते हैं, तभी आप अगली जो ट्रेनिंग है रिमोट सेंसिंग की वो समझ पाएंगे, और तभी आप अपने स्टूडेंट को प्रेरित कर पाएंगे. पिछले दिनों में, पिछले लास्ट ईयर में जो 14 स्टूडेंट्स जो अपने रिसर्च करना शुरू हो गए, वो केवल इसी ट्रेनिंग की, इसी तरह की ट्रेनिंग डॉक्टर शर्मा साहब ने अरेंज किया था, हैंड्स ऑन किया था. उसके बाद से दे आर केपेबल इनफ टू डू देयर रिसर्च प्रोजेक्ट इन ए पर्टिकुलर फील्ड. तो आई एम श्योर कि यदि आप ये करते हैं, then you can ask your student, you can sort out your students and can tell to us, and then we will teach them, we will train them, so just to start with their project work. तो इसलिए जब भी आप अपना स्टूडेंट को चाहेंगे कि वो इस रिमोट सेंसिंग में जाएं, तो आप बिल्कुल फ्री रहेंगे. हम उनकी ट्रेनिंग अलग से हैंड्स ऑन ट्रेनिंग जैसी आपकी 21 डेज की, इतनी 21 डेज की ट्रेनिंग उनकी भी करा देंगे. पर बिफोर दैट आपको भी मालूम होना चाहिए कि रिमोट सेंसिंग की शुरुआत कैसी होती है और कैसे हम उसको करें. आपको जो भी अभी शर्मा जी और उनकी टीम जो बताने वाली है, वो आपको फ्रीली अवेलेबल ओपन सोर्स सॉफ्टवेयर है, उनके ऊपर आपको ट्रेनिंग देंगे. तो आपको ये सोचने की जरूरत नहीं है कि आपके पास सॉफ्टवेयर है कि नहीं, आपके पास एसेंशियल नॉलेज है कि नहीं. आई एम डैम श्योर कि आफ्टर 21 days you will be able to start the GIS work. So I am thankful to Dean, College of Agriculture Engineering, PI, NAHE project, and the team of the Dr. Sharma, including Mr. Saurabh Nema, Ankit ji, Rawat ji, Popat ji, and all one who will be with you in all 21 days, 8 hours per day. So I'm thankful to you also that you have volunteered yourself to have the knowledge of remote sensing and GIS. So I'm thankful to you that you have come to this place, I mean for this training. and we will meet you again and again, and periodically we will meet. Our PI and Dean and everybody will be in online whenever it is possible to you. So I'm thankful to you, and just over to Dr. Sir.
Thank you very much, sir. Now I request Dr. Shivam Krishna sir to please start the presentation.
Is it audible?
Yes, sir, it's audible. Sir.
Just a minute, I am sharing my screen. Screen is visible to all of you? Whether my screen is visible to all of you?
Yes, sir, it's visible.
Okay. And sound is clear? Is there any echo?
Sound is clear, sir, but it's you can speak little bit louder, sir.
Okay. Then. Very good morning to respected Dean College of Agriculture Engineering, Principal Investigator of ICR Nahib CAAST project, Program Coordinator, and my dear colleagues who are colleagues and seniors who are participating in this program. So today I am here to present the information on the topic Introduction to Remote Sensing and its Application in Agriculture. So before starting with the remote sensing introduction, I want to discuss with you about the major heading, that is the geospatial technology. So the geospatial, because the remote sensing comes under the component of the geospatial technology, the geospatial technology is a term which was used to describe the range of all the modern tools which was contributing to the geographic mapping and analysis of the Earth surface. This technology is not new. These technologies have been evolving in some form from the early 90s, and before that it was the geospatial technology was done through the balloons and the cartography in which the camera was fixed and through which the photography was done, and with the as the time progresses, the advancement was came in this technology, and with this there can come into the existence of the geographic position system. So the geographic position system is a system which was used to pinpoint an exact location on the Earth's surface, which was possible by the vast network of the satellites that are located in space, and from that a very useful information regarding the navigation, regarding the globe position, and regarding the weather pattern, and everything will be gathered. And the next component of the geospatial technology is the geographic information system. The geographic information system is nothing but it is an amalgamation of all the softwares and the automated technologies related to the computers, which was used to capture, analyze, interpret the data which was transmitted from the navigation satellites, and through which useful information will be generated, and the targeted audience can use it in wider objectives in order to satisfy the socio-economic development of the society. So majorly the application of the remote sensing is broadly classified into three types. So first is the application of remote sensing in the precision agriculture. So the precision agriculture is the concept was normally applicable, and it is presently in wide occurrence in the European nations and the developed nations, because there the marginal land holding of the farmers is very high, and here the land holding of the farmers are marginal farmers, and the land holding is less than 1 hectare. So here it is somewhat difficult to apply and introduce this concept. So, but even though there are some those farmers which are the progressive farmers, and though some of the states, they are the farmers of some of the states, they are using this precision agriculture also. In this precision agriculture was conducted to optimize the inputs such that there will be no wastage of the input, and in such a way agriculture productivity and production can be maximized. But mostly for making this precision agriculture applicable, a large field size is required. In such a way only we can reduce the cost of the cultivation with mechanization and using all the aspects and technologies of the remote sensing. So the basically the topic on which I will discuss is the digital agriculture, because at the farm level or field level we can practice the precision agriculture, but at the field level and the village level and the block level, we have to we will use the digital platform provided by this geospatial information technology. And with this geospatial information technology, the information what we will get, we will use it in the planning of the natural resource utilization in a sustainable way, and we can optimize the farm input such that the farm income can be doubled and farmers can be benefited. And at the large level, you can say at the macro level, we can utilize this concept of the digital agriculture, and through which farming can become a profitable venture, and it can become more economical and become less power intensive. And the next is the smart agriculture, in which there will be the use of the information technology and all those Internet of Things, such as the machine learning techniques, artificial intelligence. All this we can use, and through which the farmers can take the decision on the crop management proactively, and he can make the production and the productivity of his farm more profitable. So in the upcoming slides we will discuss mostly about this digital agriculture and the smart agriculture, which will make the decision making, which will enhance the decision making power in the of the farmers. So now, in the present era of the climate change and all those problems which was reducing the farm income, the mostly we all the at the research level also, at the farm levels also, all the stakeholders are trying to optimize the utilization of the agriculture resources, particularly because the water is depleting, and all those fertilizer resources, for example the phosphate reserves are depleting. So we want to optimize the utilization of the agriculture resources, particularly the soil and the water, and such that the agriculture will be a sustainable venture. And in order to double the farmer income and reduce the farm input and enhance the agriculture productivity, our target is also to optimize the utilization of the inputs such that we can get the maximum productivity through the minimum input, like minimum fertilizer, minimum seeds. There should be the less investment on all those crop protection technologies which was used and applied for the disease and the pest management. And for making this digital agriculture a successful platform is required, and that platform is provided by this remote sensing and the geospatial information system technology, because the information which was generated from this technologies, we can manage our crop we can also manage our soil. And basically through this remote sensing and the GIS, the management is at the two level. It is first is at the soil level and second is at the crop level. Because in a very feasible manner and in a very economical and within a with less power input and we can also say within a less time we can manage we can get a good crop growth. We can manage the disease and the pest infestation. We can reduce the disease and pest infestation. We can produce more crop per drop of the water. And we can also make our agriculture ecosystem a climate resilient ecosystem by assessing the drought before it comes, or assessing the disease on or the pest incidents before it reaches to our farm's field through with the help of the remote sensing technology. And in the same way, we can also characterize the soil. We can also identify the land degradation pattern. We can also identify for which crop, which soil will be suitable. We can also identify the soil nutrient status, and all these things we can do it in a non-destructive manner, because we all of us are from the agriculture field, and all of us knows that for estimating the nutrient, for estimating the soil quality, we are doing it in a destructive manner in a field. So while doing it in a destructive manner, there will be human error, there will be environmental error will be there. So if with the help of this remote sensing technology, we can do it in a non-destructive manner, and within a small time frame, we can get a large amount of the information, which will be useful for utilizing our soil resources in a very optimal way. So first of all, remote sensing. So the remote sensing, there are three platforms. Remote sensing means sensing the object or the characterize the particles which was present in the earth surface from a remote place. This is the main definition of the remote sensing. So there are three platforms for the sensing the data from a remote area. First platform is the satellite remote sensing, in which we can sense the data through the satellite. We can characterize the object, we can identify the target object from the satellite. And second one is the by aerial platform. Aerial platform means by using the helicopter, by using the unmanned aerial vehicle, by using the drone. So this is the second classification of remote sensing data collection. So the platform here is the unmanned aerial vehicle. And the third platform is the proximal sensing. So proximal sensing means ground-based data collection. If we discuss regarding this satellite remote sensing data collection, from the historical point of view, the first remote sensing data was collected through the satellite, that is the resource set. Am I audible? Hello, am I audible? Hello.
Yes, sir. Yes, you are audible.
Okay, sir. Okay. So the first remote sensing data through the satellite was collected through the resource set 1 and 2, and the spatial resolution, that which I will discuss in the upcoming slide. What is the spatial resolution? So just for characterizing this this satellite, I want to tell that it gives the data. It gives the data in a multispectral data we will get, and the spatial resolution is 5.8 meter. And in case of the Landsat satellite, the data which we will get, the temporal resolution is 16 to 18 days. After every 16 to 18 days, we will get a set of the data for the same point. And then there will be some of the international satellite, that is the European Sentinel-2, in which we will get the 10 meter pixel of the spatial resolution for NDVI on every 10 days. So the temporal resolution is 10 days. Rapid eye constellation is there. In the same way, we will get a NIR bands we will get, and that to a very sharp spatial resolution of the 5 meter. And then there is a GeoEye-1 system where we will get a resolution of 1.84 meter. And then there is a Worldview 3, which collects the multispectral data from the RGB bands, including the red age. And through the proximal remote sensing also we can collect the collect different type of the information through by using the hyperspectral sensors and by using the multispectral sensor. So this is just an over just I want to explain you about the platforms. This is from now only I will start the basics of the remote sensing. What is the basic of the remote sensing? So remote sensing, the principle behind the remote sensing is that each and every organism which was present in the Earth surface is having a typical structural character, typical physical character, typical chemical character. No two organisms are similar. Each and every organism will be having a characteristic these features. Okay? So this feature, when the light interact with the organism, and due to due to this variation in this structural and the physical character, the interaction will be specific, and due to which the whatever the light which was reflected, or whatever the light which was emitted by that organism, it was also specific and specific to that particular object or the organism. Okay? So on that basis, whatever may be the radiation which was emitted, light which was emitted and reflected by that particular organism, that was sensed by the sensor which was present in the platform. For example, you can see here the light which was coming from the sunlight, it was reflected by this tree, and then it was sensed by the sensor which was present in the satellite, and thereafter that information was sent to the station, and where the image or the signals or the spatial signal or the temporal signal, it was generated on that basis. By making by interpreting the data, spatial data or the temporal data or the spectral data, we can analyze and we can identify what may be what that target will be. We can easily identify by interpreting those data through the images or through the all those kind of the data which was provided by this remote sensing. So the remote sensing is the science or the techniques of interpretation of reflected, absorbed and emitted radiation after interaction from the earth object. Whatever the reflected radiation will be there, that was specific to that particular object, and that was sensed. And particularly in the remote sensing system, there will be two things. First is the platform, and second one is the sensor. Platform means कुछ भी हो सकता है. It may be unmanned aerial vehicle, it may be aerial means helicopter or phenocopter, it may be satellite. Sensors, anywhere the sensors will be there. Sensors may be in the satellite, it may be in the phenocopter or the helicopter, it may be in the drone. Okay? So the sensors are also of different types: the optical sensor, thermal sensor, microwave sensor. Okay? That also I will discuss in the upcoming slide. And the platform already I had explained. Okay? So as all of us know about the dual nature of the light, that light is having a particle nature also, and light is having a wave nature also. Particle nature, due to the particle nature, light is having the energy, and due to the wave nature, light is having a wavelength and the amplitude and the frequency. Okay? So whatever may be the light, it will be having its own characteristics, wavelength, it may be its own characteristic frequency, it may be its own characteristic frequency. So these are the electromagnetic spectrum of the light. On the basis of the wavelength, it was divided. Okay? And starting from the gamma rays, which will be having a very low wavelength and high energy, X-rays, then there comes the ultraviolet, visible, infrared. So these are the characteristic electromagnetic spectrum, which ranges from the shorter wavelength to the longer wavelength. And all these lights will be having its own application in the day-to-day activity. But in the case of the remote sensing, what light, what range of the light is useful, from which we can get the information, is that first is the visible wavelength of the light. Visible spectrum which range from the 400 nanometer and to the 750 nanometer. Okay, so this from this this is a visible light which comprises of the video, all of us know that. Okay, and whatever the light which was reflected from the earth or organism, whether it will be the visible light, and thereafter it comes the infrared light with ranges from the 750 nanometer to 1 millimeter. Okay, na? So infrared light, infrared light is also of two types. First is the near infrared band, and second is the thermal infrared band. That will be also characterized on the basis of the wavelength. Like the higher wavelength will be the thermal infrared band, and the lower wavelength will be having a near infrared band. And after that, this light which is the more than 1 millimeter up to 1 meter, it will be the microwave, and thereafter it comes the radio wave. So these are the electromagnetic spectrum out of all the light which was present in the cosmic rays. These are the main electromagnetic spectrum which was useful for the for characterizing the object on the basis of the remote sensing. So not only the sensors will be able to capture the image also, and the sensors will also be able to give the data of the spectrum, give the data of the spectral reflectance, give the data of images also. So there are variety of the imaging cameras. The camera which detect the which detect the red, blue, and the green color light is the visible camera. That is also called as the RGB camera. Okay, that the camera which and this RGB camera is you can find even in all the satellite. It was fitted in all the satellite. Okay, visible camera. Then there comes the infrared camera, which captures the images of the light which captures the image of the object which reflects the infrared light. So this is an infrared camera. Okay, so which infrared, and thereafter comes the near infrared or the short wave infrared region camera, which was also called as the thermal camera. Okay, so this visible light is mainly comprises of the red, green, and the blue, and particularly the vegetation which will be having a chlorophyll content, and all the other pigments like carotenoid and anthocyanin, they will emit this light. And the near infrared light was emitted, was reflected due to the presence of the water in the tissues of the plant. Okay, if the water is more will be the infrared reflectance, water is less will be the infrared reflectance. Okay, so these are on that basis their camera or the imaging technologies have been divided and characterized. So the nowadays the advanced and the more informative information giving sensors are also present. For example, the multispectral sensors and the hyperspectral sensors. So the multispectral sensor means the camera or the sensor which will be able to detect the light of more than 5 wavelengths, which wave bands, or we can say the bandwidth, which can detect the light of more than 4 or more than 5 bandwidth. Okay, na? It may be, for example, here you can see that जो camera है multispectral camera, it is detecting the 5 wave band, that is the comes under the visible wave band, and one comes under the infrared wave band. So this kind of the sensors are also there. These are the multi-dimensional sensor. If a platform is having this kind of sensor, you can gather multiple information, and what information we will get, that I will explain you in the near future. So the next comes the hyperspectral sensors. Hyperspectral sensor, you can find that here the sensor will be able to detect more than 100 bands, more than 100 and large number of the bands it can detect, and that too the resolution of less than 2 nanometer or 2 to 20 nanometer. The differences of the wave band which can be detected by this sensor is having a difference of uh, 2 is having a resolution of 2 to 20 nanometer. So with this hyperspectral sensor, even though it is very costly, but this hyperspectral sensors will be able to characterize any object in a multiple way. We can gather a multiple information. We can gather we can characterize any object. For example, if a wheat plant is there, we can characterize the wheat plant on the basis of chlorophyll. If we are using the hyperspectral sensor, on the basis of chlorophyll, on the basis of the biophysical parameter, just like lignin, protein, carbohydrates, sugars, everything we can detect in a non-destructive manner. So now I had ended up with the platform which was used, sensors which was used. Mane ki jo I had end up with the infrastructure, the major infrastructure or the major structure of the satellite, or the major structure of the ground-based remote sensing satellite, or the aerial-based remote sensing satellite that we had completed. Now, what are the type of the data which we can generate with the help of the remote sensing? That are of 4 types. First is the spectral resolution, spectral data. Second is the radiometric data. Third one is the spatial data, and fourth one is the temporal data. Okay, so this 4 kind of the data, we can characterize the remote sensing data on the basis of this 4 categories. Okay, so I can explain it one by one. First is the spatial resolution. Spatial resolution means the minimum size which you can detect. Minimum it is defined by minimum size of the pixel which represents the area on the ground. And in this kind of the if the sensor will be having a smaller footprint, tends to have high spatial resolution, and the sensor with large footprints tends to have low spatial resolution. For example, if you are seeing here, you will find that here the picture is very clear. Here the picture is very clear. Here the picture is very clear. Why? Because the pixel the spatial resolution is very high, and the pixel is a very smaller footprint of the pixel is there. Okay. Or here, if you find that we can find somewhat the image which was hazy. Why? Because the pixel size is very large, and here also you will find that it was the pixel size was very large. Okay, na? So what is this pixel? Pixel is that it represents the area. If an area is of 1 kilometer by 1 kilometer, so the pixel means the minimum ground area which can be captured by the camera or the sensor. Minimum. Here if you find that if this sensor is able to capture the 10 meter area, 10 meter by 10 meter area it is capturing, it is having a very fine resolution. On here, if you are finding that this camera will be able to capture a area of 50 meter by 50 meter. So that means here the spatial resolution is medium, and here you will find that in this black area which was given, you will find that it was able to capture an area of 100 meter by 100 meter. So this will give a very coarse picture. This will give a very hazy picture, just like in the previous slide you will see. So lesser the footprint, smaller the footprint, more will be the spatial resolution, and larger the footprint, it will be the lower will be the spatial resolution. So size of the pixel. Size of the pixel means less than 10 meter, fine resolution, pixel size 50 meter into 50 meter, medium resolution, and pixel size 100 meter into 100 meter, coarse resolution. So if the pixel size is less, you will get a very good picture. In some of the application, you will find that while searching the Google engine, you will find that you are getting a very clear picture from some of the application software, from some of the GIS software, you will getting a very coarse picture. So this is the reason behind that is that is the sensors which that satellite is using, it will be having a very coarse resolution, which will be giving the very hazy picture, and the finer resolution giving the more clear picture of the object. Here in this picture, you will find that in this satellite picture of of an area which will be having a community settlement also, which will be having a agriculture also, field also, which will be having an everything, road is also there, and the water is also there. You will find that the picture is very clear. Even you can able to clear, or you can able to distinguish the field boundaries is also very clear. So because the resolution is 5.8 meter here, and in this picture, the next picture is that where the spatial resolution is 23 meter, you will find that the picture is very hazy. That you can even don't find the no field boundaries also is not detectable. So that means this satellite is having a very low spatial resolution, and in this picture you can even not find the even you will not able to distinguish where is the community settlement, where is the field. So no boundaries, nothing, because the 56 meter spatial resolution is there. So higher the footprint, lower will be the spatial resolution. So next is the spectral resolution. Spectral resolution means spectral data. It is one of the form of the data which we will get from the remote sensing on the basis of how many bands it can, how many wavelength of the light it can recognize, it can detect, it can sense. That sensor can sense. So on that basis, we will get information about that object. For example, here the whatever the spectral signatures which you are getting here, on that basis we can say that this sensor, this remote sensing is giving a spectral data of 6 bands, 0.5 micrometer to 1 micrometer. It will be able to detect the 4 bands, and in between the 1 micrometer wavelength to 2 micrometer wavelength, it will be able to detect the 1 band, and in between above the 2 micrometer, it will be able to detect the 6 band. So that means that it is a kind of multispectral sensor. ठीक है ना? Just a minute. Hello. सर लेक्चर चल रहा है सर, 21 डेज की ट्रेनिंग है अपने नाहिद की, उसी में लेक्चर चल रहा है. हाँ. ओके सर, ओके. हाँ. ओके सर. हाँ. थैंक यू सर. Okay, some urgent call is there, so that's why some interruption. So in this way we can also so the remote sensing data can be also gathered, will also give you information about the spectral wavelength. Just a minute. So the next type of the data which was provided by the remote sensing satellite and the platform is the temporal resolution. Temporal resolution means for satellite, it is the time taken to complete an orbit and revisit the same observation area. जैसे कि अभी एक सैटेलाइट अगर JNKVV के ऊपर, JNKVV को अगर मान लो आज 8 बजे दिखा रहा है, तो वो उसी JNKVV को कब ऑब्जर्व करेगा, वो टाइम पीरियड कितना रहेगा, कितना टाइम लगता है उसको सेम माने एक ऑर्बिट में घूम के आके and to revisit the same observation area. So this is this type of the data we can also call it as the temporal data, and the lesser the temporal lesser the days required for completing an orbit, the more information we will get. For example, for completing an entire orbit cycle and coming back to the exact same area, Landsat requires 16 days. Landsat needs 16 days. The Modis software needs 1 day, and Nexrad needs 6 minutes. Okay, and so in this way, the temporal data, the remote sensing data can be characterized on the basis of temporal resolution. Now, the sensors will also be able to capture the images because the cameras are also there. Already I had explained some sensors will be having the RGB camera, some sensors will be having a thermal camera. So how that image will be interpreted. So whatever may be the target, that target reflect some of the light, and that light, when it was interacted with the sensors, it ultimately ultimately an image will be formed, and that image, after processing with the help of the automated softwares and the technology, one can interpret and analyze the object. Here, with this diagram, I want to explain you. For example, this is a remote sensing satellite list 3. In this remote sensing satellite, you will find that there are 4 kind of the cameras are there. For example, this B2 this B3 camera will able to detect the green bands, green wavelength band. This B4 will able to detect the red wavelength band. This B5 will be able to detect the infrared wavelength band. And when all these colors when was mixed in a single platform with the help of the computational software, we will get an image like this. Okay, we can get an image like this after merging all those 3 wavelength bands. Okay, we will get an image of an object like this. So for analyzing an object, for interpreting and gaining some insight about the object, and in order to characterize the object, one should have a knowledge about all this kind of the remote sensing data, characteristics, spectral data, spatial data, temporal data, radiometric data. All those kind of the data we have to we should have to and able to analyze and interpret some information about the target object. So so with this the remote sensing data characterization was also, and how many type of the data we can get from the remote sensing. Now the next topic is the spectral signature or the spectral reflectance curve. So why this topic is important? Because each and if there are 2 human beings, for example, Ram and Shyam, both Ram and Shyam will be having his own individual signature. उनका जो सिग्नेचर है वो सेम नहीं होगा. They will be having their own characteristic signature. ठीक है ना? So in the same way, each and every object which was present in the earth surface, it will be having its own signature. It will be having its own spectral signature. Okay. So which way, and that information about the spectral signature will be gathered with the help of by studying the spectral reflectance. So the spectral reflectance means is a graph of the spectral reflectance of an object, is a function of wavelength, and is very useful for choosing the wavelength region for remotely sensed data acquisition for certain applications. For example, water bodies. Here you can see the water bodies, which is a clear water. It will be having its own spectral signature. When it is reflecting, it is reflecting a light of 0.6 micrometer. 0.6 micrometer, the reflectance is maximum. In case of the vegetation, the reflectance is maximum at the green and the green region, that is the between the 0.5, 0.4 and 0.6 green region. All of us know that the plant is reflecting the green light, that's why it is looking like green, because chlorophyll is present. Chlorophyll reflects the green light. And the infrared light was also reflected maximum by this vegetation, which is green vegetation, and particularly the dry vegetation will also reflect this infrared light in a maximum way, in a maximum amount. So you can also see here the dry soil is also having its own characteristics, spectral signature. Okay? Spectral signature, it is because here in the x-axis you will find that it is wavelength is there, and in the y-axis reflectance is there. So what kind of the light an object is reflecting, that information will also be gathered with this graph. and what amount of the light was reflected, that also will be gathered by this graph. Okay? So the spectral reflectance is very much essential if you want to characterize any object. Okay? It will be having a large amount of the application. For example, it is useful for identifying the object which was present in the earth feature. It is also useful for identifying the characteristic spectral wavelength of the light which was reflected maximum by a particular object. For example, a wheat crop is there. For example, a tomato crop is there. So if a particular tomato crop is reflecting a light of 0.66 micrometer of the wavelength, that is and it is reflecting in a maximum amount, that should be the characteristics of that particular tomato. That is the spectral signature of that particular tomato crop. So if we want to detect the area which was occupied by that particular tomato crop, so we can use in a maximum area, in a large area at the community level or in a state level, we can, in the satellite, we can use that kind of sensor which it can sense 0.66 micrometer of the wavelength. So in that way, we can able to detect the tomato crop which was grown in a
Hello,
particular area.
excuse me.
Hello.
Hello.
Yes, sir.
Issue with
Hello.
voice is clear and
Sir, voice is not clear. Sir, my voice is clear.
हाँ, वो
all
इंटरनेट के
the
इशू के चलते रुक के रुक के आ रही है, बहुत रुक के आवाज आ रही है, हम कलेक्ट ही नहीं कर पा रहे हैं कि आप क्या बताना चाह रहे हैं.
So, sir, what we will do, because this is the platform through which we are I am taking the, यही एक वेबएक्स के थ्रू इनने लिंक शेयर किया था, आप लोग भी सब वेबएक्स से ही जुड़े हुए हैं. Now it is
नाहिब
clear?
में जूम भी लाइसेंस वर्जन अवेलेबल है, यदि जूम में कराई जाए तो क्या दिक्कत है?
हमारा सजेशन भी यही है, या तो
वेबएक्स जो है लो बैंडविड्थ पे बहुत प्रॉब्लम आती है.
भले Zoom कुछ नहीं आ रहा है. अच्छा
इवन
है
सरहद की
कि
आवाज भी क्लियर नहीं आ रही, ब्रेक हो रही.
पूछा आपको.
So just a minute, I will contact with the organizer.
रामकृष्ण प्लीज, यदि सौरभ, डॉक्टर सौरभ या नेमा सर तो शायद दूसरी मीटिंग में बिजी हैं, थोड़ा कंफर्म करिए ना यदि जूम पे पॉसिबल हो तो.
हाँ, जूम पे या अपना उस पे अभी गूगल पे कर लेते हैं, फिर लंच सेशन में या बाद में डिस्कस करके फिर से देख लेंगे क्या इशू अपन रेज कर सकते हैं, क्या रिजॉल्व कर सकते हैं. 15 मिनट का वेट अपन जूम पे.
सर अभी इसमें कंटिन्यू
ठीक.
आप लोगों की वॉइस क्लियर है सर, विशेन सर और शर्मा सर, आप लोगों की वॉइस क्लियर है, केवल वहीं से प्रॉब्लम जा रहा है. रामाकृष्ण सर की वॉइस में कहीं वहां पे कोई इशू है.
आउट कैंपस का इंटरनेट कनेक्शन ज्यादा अच्छा है ना.
नहीं, पार्टिसिपेंट की आवाज आ रही है, स्पीकर की आवाज नहीं आ रही है.
स्पीकर का आवाज तो आ रही है सर.
आपको
वॉइस क्लियर नहीं
सबको
है,
ये प्रॉब्लम जा रही है?
नहीं सर, वो सॉफ्टवेयर
सबको
आप
ये प्रॉब्लम
डाउनलोड करके देख लीजिए, इंस्टॉल करके उसमें से लगाइए, सही रहेगा.
एक मिनट सर, एक मिनट. मैं मोबाइल से जुड़ता हूं सर, मोबाइल से कनेक्ट करता हूं, जस्ट अ मिनट.
The difference of the sum of the difference.
Sir, I am just joining with the mobile, so please wait. I am just joining with my mobile.
The difference of the sum of अपने difference की spelling. Sir, अभी इसमें कंटिन्यू चल रहा है, इसमें कंटिन्यू चल रहा है. Difference की spelling. थोड़ा संभाल के लिखो कि यानी तुम्हारा कितना गंदा बना है। डिफरेंस ऑफ बोलो। हाँ
Hello, sir. Now it is audible.
क्या कर रहे हो?
Now it is audible.
Now it is perfect. Perfect.
Okay. Just a minute, sir. Actually, I joined with my mobile now.
Yeah. Now voice is very clear.
Sir, slide is visible. Slide is visible.
Yes, sir. Very visible.
Okay, sir. So this is the so on the basis of the spectral signature, we will be able to characterize that particular object कि उसका spectral signature कैसा होगा, which wavelength of the light it was reflecting maximum, and on that basis we will use in a broad way by using that particular sensor which can sense that particular object using it in a satellite, such that we can we can identify that particular crop or that particular object in a maximum area. For example, if the characteristic spectral signature of the tomato is there, it has having maximum reflectance at 1 micrometer. So if we will use that kind of the sensor in the satellite, we can identify the area which was covered by the tomato in a particular state in a particular area. Okay, so that with this kind of information will help in planning a particular crop studying a crop inventory in planning, in the land use planning. In land use planning also we can use, even we can also use that information in which area that crop is growing good, having good health, having good productivity on the basis of the vegetation NDVI, which I will explain in the next slide. So here in this spectral signature also you will find that the citrus, tomato, sorghum and cotton, each will be having its own spectral signature. On that basis that plant will be characterized. And you will find that this crop at the particular stage will show a specific spectral signature, and in the, for example, in the vegetative stage mein the spectral signature will be different, and in the reproductive stage the vegetative the spectral signature will be different. So but the experts told that in order to estimate the spectral signature, the ideal condition is that before flowering or at the time of the flowering we have to take the spectral signature data. So so how this spectral signature was generated, because the plant cell is having a chlorophyll in the mesophyll cell, and that chlorophyll will reflecting this red light and in red light and the red and the blue light and the water which was present in the mesophyll tissues, that reflects the infrared light. Okay, so on that basis the healthy plant which will be having a high chlorophyll will reflect a large near infrared light, and that reflectance pattern or the spectral signature will also varies on the basis of the composition of the cell. For example, on the basis of the chlorophyll content, more the chlorophyll content, more will be the reflectance of the near infrared, internal structure, lignin, cellulose, water content. If the crop is facing any crop stress, if the crop is facing nutrient stress or water stress, the spectral signature will vary. And if the crop is having, if someone is vegetative stage, then the spectral signature is different. If it is at the reproductive stage, the spectral signature will be different. So so how we can estimate about the crop health, how the health of the crop is, whether the crop is facing any stress or not, particularly abiotic or the biotic stress or not. So that was identified on the basis of the emergence of concept of vegetation indices.
So the vegetation indices is nothing but the ratio of the whatever the infrared light which was reflected and to the red wavelength light which was reflected, and it is an excellent measure of the vegetation health. Okay. Healthy crop, what it will, healthy crop will reflect more infrared light and green light. Healthy crop will reflect, the reflectance of the green light and near infrared light is very high, and the non-healthy crop or the stressed crop will reflect more blue light and the red light, because all of us know the key for the photosynthesis blue and red light is necessary. So the healthy crop will not reflect. Healthy crop will reflect only green and the near infrared light. Or the stressed and the damaged crop, why it will reflect the blue and red light? Because it will be having some decrease in the chlorophyll content, it will be having some of the changes in the internal leaf structure. Okay. So for in this way we can able to detect the crop stress, whether the crop is having any nutrient stress or the water stress or the biotic stress. Okay. So next is the formula. What is the formula of the vegetation spectral index or the normalized differential vegetation index? It is the formula is near infrared wavelength minus red divided by near infrared plus red. Okay. On that basis we can assess the chlorophyll content. If we will develop an equation of the regression on that basis, we will assess the chlorophyll content and we will assess the lignin content. We will assess the membrane stability. We will assess the type of the crop, what kind of crop is there, nutrient content, biomass, crop yield. So all these kind of the parameters we can identify. So the NDVI value range from the 0 to 1. If 1 means it is a very healthy plant, 0.66 to 1 means it is a very healthy. Value is in the minus, it will be the dead plant or the object is of non-living nature, or the some bare rock will be there. So now I will come in the application part. All the basic part I had completed. Now I will come in the application part. So this the application of the remote sensing is the crop classification. Means ki preparation of the inventory ki what is the acreage of the wheat crop, rice crop in a particular area, or the different crop in a particular area, acreage estimation. Then the yield modeling and the estimation. What are the how we can also forecast the yield also. On the basis of the remote sensing data, we can identify the crop phenology ki what kaun se stage mein hai, vegetative mein hai, reproductive mein hai. We can also identify that crop phenology. We can also identify the crop conditioning, whether the crop is at the whether the crop is facing some drought, or the it can also detect the give the information about the stress, whether the crop is facing any abiotic stress or the biotic stress. So in this way, all this kind of the information what we are getting on that basis, and the remote sensing is having application. So this is one of the research paper which was research conducted by the presently the director of the ISRO, that is the Suresh Kumar, in 2019, and he has given the classify the crop in a particular Saharanpur district, given the land cover or the land use of that particular district, and he identified the different crop area, wheat, sugarcane, orchard, fallow land, forest, all those kind of the ecosystem, agroforestry ecosystem he has defined in this particular area through the digital image processing. And this has been done through the generation of the spectral signature. Because before that we have to identify what will be the spectral signature of the wheat crop, what will be the spectral signature of the sugarcane crop. Then only we will be able to generate the signature. Then only we will be able to generate the data of the crop classification or the crop inventory map we will able to design. And this is the NDVI same for the same district. NDVI of wheat crop was identified. You will find that in this area the NDVI is around the 0.9. That means the area the crop which was grown in this area is very healthy and having more NDVI, and they facing no stress or the or some good crop condition is there. But in that particular area, in this area, you will find that the crop condition not good, because it is a specific NDVI of the wheat. Other crops are not there. So this yellow area means the crop is facing some stress, or the crop is not in the good condition. On that, so this on that basis we can identify the crop condition, in which condition the crop is. So the next application is the crop yield modeling, and we can also identify or forecast the yield for the particular crop when it is at the vegetative stage or the reproductive stage. So this was done on the basis of the evaluation of the NDVI. At the evaluation of the NDVI at the different plots at the different stages, and multiple regression equation has been initiated, which is designed. On that basis, an equation will come. So for that particular area, with the help of this equation, we can calculate the yield. Again, and whatever may be the predicted yield and the predicted yield and the observed yield, that can also be correlated, and a high correlation was found when it was done with the when NDVI was used as a cofactor. So in this picture, you will find that they are also doing the ground truthing in this area of 1 meter square. They are also doing the ground truthing. They are also they are also predicting the true yield, and for the preparation of the yield model. So so on that basis, we can also forecast the yield, and we can prepare the crop yield map also. We on in which area area specific crop yield map will be prepared, which area the crop yield is more, which area the crop yield is less. So that will also having some application in the insurance sector. So these are the some of the recent vegetation indices already, which I had explained. That is the NDVI. The NDVI, apart from that, there is the soil adjusted vegetation index, the modified soil vegetation index is there. So green normalized difference vegetation index. So here it is a very less number of the indices was mentioned, but there is more than 50 to more than 35 to 40 indices is presently people are using. They had generated the information and for estimating the for interpreting the biophysical nature of the object, and the for deriving the information of the object. So the here the crop condition was assessed. The assessment of the crop condition, whether the crop is facing some kind of the stress or some kind of abiotic or biotic stress, that was done through the use of the spectral vegetation indices. In this, the vegetation indices at the near infrared and infrared in case of the NDVI was taken. In case of the vegetation condition indices, the maximum NDVI and minimum NDVI was taken. In case of the temperature condition indices, the maximum we can also estimate the temperature with the help of the sensors also. So on that basis, the temperature condition was also estimate for even it is also universally known fact that when the crop is facing some stress, particularly the drought stress or the high temperature stress, high temperature stress, the temperature will be high. The plant will not be able to maintain the heat balance because of the less transpiration. Stomata close ho jayega. There will be no transpiration. So the heat stress will face and temperature will increase. So on that basis, the temperature will be sensed by the sensor, and temperature condition index will show the stress. Will show the stress levels faced by the crop. So you can find that the crop water stress indices, the red area denotes the crop is under high stress, and the green area denotes the crop is under the less stress. Okay, so in that way, on that basis, this is a thermal image of the thermal camera. The thermal camera and thermal sensors was used in that UAV. And this thermal imaging is which is usually and commonly used for the detecting the abiotic stresses, particularly the high temperature stresses. And in such a way, the developed countries where this technology they are using, they will be able to manage the irrigation. They will be able to schedule the irrigation. in an optimum and the in an optimum and the sustainable way, conserving the resources. So the crop water stress is related to the leaf temperature, and the leaf temperature was with the help of leaf temperature we can also estimate the crop water stress index, and crop water stress index will gives the idea of the abiotic stress, particularly high temperature and drought faced by the crop. So in this way, with the help of the remote sensing technology, a crop water stress will be assessed. So now the next is the crop pest infestation. Whenever a pest will infest in a crop, some kind of the in plant also we can find that some yellowing or the necrosis kind of system, chlorosis kind of the symptoms will appear. So due to which the chlorophyll content reduced. So which changes the spectral reflectance pattern. So by studying this spectral reflectance pattern, it was found that the healthy plant will reflect more near infrared light, and the stressed plant will reflect less near infrared light. So this kind of change in the reflectance pattern will takes place 7 to 10 days before the symptoms appear. So if we can know about the insect infestation 7 to 10 days before, we can schedule the pesticide application and in a proactive manner, and we can protect our yield. So presently the spectral the hyperspectral sensors will only for this in case of the biotic stress, because during the biotic stress there will be some minute changes in between the biotic and the abiotic stress. Now, if the crop is facing both biotic and abiotic stress, then we will not be able to distinguish whether with the help of the spectral data whether the crop is facing biotic or abiotic. So in order to differentiate it, we have to use the hyperspectral sensor, because in that sensor we can get more than one band of the narrow bandwidth, and we can gather large number of the information. And there are presently also this research is in progress in order to characterize the diseases, and less number of the work was done in case of the diseases as compared to the abiotic stresses. For example, this work I had taken from the journal Sensors published in 2020, in which it was observed that the field which was infected by the rust in case of the wheat, it was the camera, near infrared camera, that is the thermal camera images was found in the images was found in the area where we can get the red color. It was for that area will be infected with the wheat rust, and in case of the healthy plant we will observe it is green. In case of the non-healthy plant, we will find that it will the color will getting the transition from the yellow to the red color. So with the help of the spectrum also, if you will observe the spectral signature, you will find that the healthy plant will reflecting more infrared radiation, near infrared radiation, and the non-healthy plant where the disease was there the reflectance is not maximum, and the spectral signature is also different in as compared to that of the healthy plant. So in this way we can also define or characterize the degree of the diseases in the plant with the help of the remote sensing data. So here in this work was published by the Indian author in 2011, in which the late blight of the potato was characterized, and it was found that the potato field which was disease infested on the basis of the hyperspectral imaging, the most the healthy plant will be having the spectral reflectance maximum at the near infrared region, and the non-healthy plant which will be infected with the late blight will be having the lesser reflectance in the near infrared region. So nowadays the thermal imaging or the thermal imaging sensors which will be able to image the temperature of the which will characterize the temperature of the object, and which will also able to capture the spectral images of the on the basis of the temperature that was came into existence, and on that basis we will exploit the change in the surface temperature due to the stress symptoms, and we will be able to detect biotic and the abiotic stresses. So, in near future, it will be observed that with the help of these very advanced sensors like multispectral sensors and the hyperspectral sensors, we can able to estimate the biophysical parameter also. Biophysical parameter which we will normally use destructive method in the lab, particularly for the leaf protein, chlorophyll, carotenoid, anthocyanin, lignin, cellulose, sugars. For all these for all these things we are conducting in lab, non-destructive manner. Maximum error is there, and environmental error is there, human error is there. So this kind of the multispectral sensors, we can characterize the biophysical parameters also through the remote sensing. That will help in the great deal. And also the different crop will also be having some different characteristic spectral signature, and that will also helps in the crop detection or the identification of the particular crop. So now I will discuss something about the work which we are doing in our NAHEP CAAST project. Is that we are doing the high throughput phenotyping with the help of this remote sensing instrument. So because in order to assist the genomic data, the perfect phenotyping is very much required, and if we will having no perfect phenotyping, then our genotype or the QTL, during the identification of the QTL, it will end up with the false QTL, and we will get the false QTL, and the whole energy of the team and the time will be wasted. In order to conserve that, we have to do the precise phenotyping, and that is in practice at the different institute in India, also in IARI, in National Abiotic Stress Management Institute, in US, Bangalore, everywhere the phenomics facility is there. So there they are just characterizing the genotypes on the basis of the abiotic stress tolerance. They are identifying the tolerant, drought tolerant, or the heat temperature tolerant genotype, and on that basis they are identifying the genes responsible, and that genes will be used in a reverse genetics manner to develop a drought tolerant or the heat tolerant phenotype. So this is used in the breeding for developing the improved genotypes. And in case of visible light, the visible camera, that is the RGB camera, we can also identify the area of the plant. For example, in this case, a shoot of a seedling of the maize area was identified, and with the help of this visible light imaging, we can also retrieve the leaf area of different plant canopy of the over the agriculture crop, and that was published in Agriculture and Forest Meteorology. So with the help of this RGB camera, we can identify the leaf area, and for the identification of the leaf area, we have to do the ground truthing with the ground truthing, and that for that the leaf area was estimated with the help of leaf area meter, and then the leaf area index, which was estimated from the digital photo. The regression equation was created. On that basis, we can standardize this technology for the estimation of the leaf area index. And these are some of the work which was done and published in some good journal, in which they had used the thermal imaging camera for the visualization of infrared radiation, which detect on that basis we can detect the temperature of the object, and we can identify which crop is facing the having high temperature, or which crop is having the good health. We can characterize in that way. In this work, they had applied the thermal imaging concept, and they had identified the water stress tolerant maize genotype with the help of the thermal imaging camera and the sensors. And the platform which was used is the fixed platform they had used. So this is a very advanced work which was conducted through this hyperspectral imaging, in which they are predicting the protein in the single wheat kernel. the protein estimation with the help of the where you doing it with the biurate method and with the with the help of the Geldal method and all these method which we are using, it is very time consuming, very power consuming. So in order to reduce the time and the efficiency, this hyperspectral data can be validated. Hyperspectral sensors can be used, and the spectral data can be validated against the protein. And it was a regression model was produced, which was done by the workers, and they are able to estimate the protein in the single wheat kernel. In the same way, in the grain protein content also, the protein content was also identified in case of the rice. For that, they had used the NDSI, and they in the advanced way they had also identified the loci or the candidate genes which was responsible for that particular normalized differential stress index. So the people have also, workers have also used this multispectral imaging technology with the help of the unmanned aerial vehicle. They had phenotyped the maize genotype. Because earlier also I went through the discussion with Dr. Vikram Gaur sir. He was also doing some work on the rice. He can also utilize this kind of the drone which we are purchasing, or they can also use these multispectral sensors which we are purchasing for the phenotyping of the rice varieties. So this paper will be helpful, and this kind of work also we can do. So this is the work which was published by Vishwanathan Chinnaswamy at IRI in 2019, in which with the help of the multispectral imaging sensor, they had identified the nitrogen use efficient genotype. They had identified the water deficit stress tolerant genotype. So with this we can also identify the nutrient tolerant genotype also, and drought tolerant genotype also. We can, because you in this RGB camera, you can see here it is the RGB camera, and here it is the thermal sensors which they had used. So you can find that the blue area, where the blue area is there the crop health is good. Crop health is fine, and where the area is yellow, or where the area is yellow or the purple in color, there the health condition is not in good shape, and the plant is facing some stress. On that basis, we can also identify these drought tolerant and the nitrogen use efficient genotypes of the rice. So this is an emerging technology in which the fluorescence light which was emitted during the photochemistry process. As we all know that during the excitation of the chlorophyll from its ground state to the excited state, and while coming to the ground state from the excited state, some of the energy which was released in the form of the fluorescence, also some energy was utilized in the form of the photochemistry, which will derive the photochemical processes, and parallelly the fluorescence light was also emitted. So it was universally universal concept that more the fluorescence light, more will be the more healthy will be the photosynthetic health of the plant. So with the help of this imaging the fluorescence light, we can also identify the photosynthetic health of the plant, photochemistry of the plant, whether the plant will be able to photosynthesize better. We can also identify the genotypes which will be having good photosynthesis. So this kind of the camera was also installed in the Nanaji Deshmukh Phenomic Research Center at IRI. So with the help of this different kind of the sensor, we can do the phenotyping for the biomass, plant structure, and leaf health, phenology. With the help of the RGB camera, with the help of the near infrared imaging and far infrared imaging, we can measure the tissue and the soil water content. Leaf temperature also we can measure. With the help of the fluorescence imaging, we can measure the physiological state of the photosynthetic machinery. With the help of this near infrared camera, temperature can also be estimated, leaf temperature, which was already explained. So these are the different type of the phenotyping platform. First is the aerial phenotyping platform. Next one is the ground-based phenotyping platform. Third one is the controlled environment phenotyping. Controlled environment phenotyping is the phenomics facility ground-based phenotyping means proximal remote sensing, where we will not use any platform. We will use the handheld or the fixed kind of the sensors we can use. For example, green seeker, for example, multispectral radiometer, spectral radiometer, and for the aerial phenotyping, we can use go for the unmanned aerial vehicle phenocopter. For example, here in case of the aerial phenotyping, a phenocopter was used for characterizing or the phenotyping the rice genotype. Some authors are also using this phenocopter, although it is very costly, but nowadays the drone which was coming, it is very cost effective. We can use the drone also for capturing the images and the spectral data. So this is the research area which we are focusing. I already explained we are doing the non-destructive phenotyping because of the poor result we are getting in the destructive phenotyping. We want to do the high throughput phenotyping, and the high throughput phenotyping was based on the principle of remote sensing technology. And the work plan associated with my work is we are classifying the wheat genotypes on the basis of the spectral signatures. We are also classifying the chickpea genotypes on the basis of the spectral signature. We are classifying the wheat genotypes for the high temperature stress tolerance. We are also classifying the different kind of the plant growth regulators for their efficacy against the high temperature stress tolerance in case of the chickpea. And what outcome we want to expect through this project is we want to develop the skilled manpower which will be able to use the remote sensing based high throughput phenotyping instrument, and who can serve at the national and the international level. We can also we want to develop a non-destructive phenotyping infrastructure in our university. So we are thriving for that. We are working for that in this program. So this will. So these are the main objective of our NAHEP CAAST project. It is to establish a center of advanced agriculture science and technology under national NAHEP project, and we want to develop the skill to use spatial data for natural resource management in agriculture. Our objective is to build basic capacity for using remote sensing and GIS techniques applied for betterment of the natural resource management, particularly in agriculture and allied sector, to identify the appropriate techniques for integration of spatial and ground data to realize problems related to land, water, and vegetation. We want to develop the user-friendly spatial products which we can patent, which we can license, which we can commercialize, and identify the technology for the policymaker, researchers, field workers, and farmers. So this is a work published by the Hapauche in a review paper in the Trends in Biotechnology, in which he has given the future of the automated phenomics, genomics, big data, and artificial intelligence in plant breeding. Here you will find that the crops are in the field, and with the help of the sensors, we are estimating the soil moisture, soil temperature, and on one hand, with the help of the unmanned aerial vehicle-based proximal remote sensing is going, and we are estimating the data on different biophysical phenomena. With the help of the satellite, also we are also identifying the we are also characterizing the crop, or we are also characterizing our treatments which we are taking. And on the other hand, a farmer can also getting a prior information in a proactive way to schedule the irrigation in a particular area which was infested by the pest, or the drought is there, or where the problem of the irrigation is there. He can schedule, he can program, and with the help of the computer and the automated technology, and in the same area the pesticide can be applied and the irrigation can be scheduled. So with this, I want to conclude my topic and the lecture, and now the lecture is open for discussion. Saurabh? Saurabh?
Yes, sir.
हाँ, सौरभ लेक्चर हो गया है.
सर एक
अब आप
प्री
बोल-
असेसमेंट ट्रेनिंग का कराना था टेस्ट, तो सर मैं लिंक
हाँ।
भेज दूं क्या ग्रुप में?
हाँ, ग्रुप में लिंक भेज दीजिए आप।
जी
शिवराम सर।
हाँ सर, हाँ सर।
सर I have a question actually.
हाँ सर, विक्रम
ये
सर
जो
बताइए।
ये जो सर सैटेलाइट्स है, ये जो ड्रोन्स आपने बताए हैं, ये जितने हैं सर, ये सर लाइट थ्रो करते हैं, फिर ये मतलब उनके जो रिफ्लेक्टेंस को ये मतलब एनालिसिस करता है या रिकॉर्ड करता है, या जो भी नेचुरल लाइट फॉल हो रही है उसको ही एब्जॉर्ब करता है ये?
सर दो प्रकार का रिमोट सेंसिंग है सर। एक है एक्टिव रिमोट सेंसिंग, पैसिव रिमोट सेंसिंग, जिसमें आप पैसिव रिमोट सेंसिंग के भी कुछ इंस्ट्रूमेंट हैं और मोस्टली पैसिव ही होते हैं, जिसमें कि सनलाइट से जो लाइट आती है और वो जो रिफ्लेक्ट होती है उसको वो कैप्चर करके डेटा जनरेट करता है। और जो थर्मल सेंसर और जो थर्मल इमेजिंग सेंसर वगैरह जो अपन यूज करते हैं उसमें एक्टिव लाइट यूज होती है। माने दे आर फॉलोइंग द प्रिंसिपल ऑफ एक्टिव रिमोट सेंसिंग। और वहां पे हम जो है जिस लाइट का रिफ्लेक्टेंस अपन चाहते हैं उसी लाइट को हम लोग क्या करते हैं, वी आर प्रोवाइडिंग दैट लाइट ओवर द ऑब्जेक्ट।
ओके सर, एक क्वेश्चन
मोस्टली
और
ऑल द
सर।
इंस्ट्रूमेंट्स आर फॉलोइंग पैसिव रिमोट सेंसिंग।
ओके सर, थैंक यू सर। वन मोर क्वेश्चन सर। जिस तरह से ये इल्यूमिना का जो एनजीएस डेटा जनरेट होता है और ये जो माइक्रोवेव का जो होता है सर, जिस तरह से इमेज कैप्चरिंग होती है सर, तो इसमें भी सेम वैसे ही होता है सर, इमेज कैप्चरिंग और डेटा, क्योंकि उस वाले केस के अंदर डेटा जनरेट होता है टेराबाइट्स के फॉर्म के अंदर और उसको बाय हाई थ्रूपुट कंप्यूटर प्रोसेस करता है सर डेटा को। तो इसमें भी सर कुछ ऐसा ही होता है सर, और उसके लिए पर्टिकुलर सॉफ्टवेयर भी होते हैं और जिसके लिए बहुत ही ट्रेंड आदमी होना चाहिए उसको प्रोसेस करने के लिए। उसके बाद डेटा फिल्टर वगैरह की
सर
जो आप दिखा रहे थे सर,
सर
इमेज वो
आपने
निकलता है
बिल्कुल
सर।
सही कहा सर। आपने बिल्कुल सही कहा सर। इसमें जो इमेजिंग के सॉफ्टवेयर हैं, जो इमेज का जो डेटा आता है उसको वो जो ग्राउंड में जो स्टेशंस होते हैं वहां पे कंप्यूटर्स होते हैं, तो वो कंप्यूटर का जो ग्रामोमेट्रिक में जिनका स्पेशलाइजेशन होता है या जिनको कि उस सॉफ्टवेयर की ट्रेनिंग ली हो, जिसको वो सॉफ्टवेयर चलाना आता हो, वही मतलब कि उसके उस इमेज को डेवलप कर सकता है और इंटरप्रिटेशन निकाल सकता है। सो वी शुड हैव सम नॉलेज ऑफ द फोटोग्रामेट्री also, and we should have knowledge of the softwares which was used for the image analysis and image interpretation. इसमें भी सेम प्रिंसिपल है, सॉफ्टवेयर्स इसमें है सर।
थैंक यू सर। सर एक और क्वेश्चन है सर। एक स्लाइड के अंदर आपने एक ग्राफ दिखाया था जिसके अंदर वेवलेंथ को प्लॉट किया गया था रिफ्लेक्टेंस से, और जैसे जैसे वेवलेंथ बढ़ रही थी तो रिफ्लेक्टेंस में वेरिएशन था। तो जैसे हम लोग एंजाइमेटिक में रिएक्शंस को स्टडी करने के लिए करते हैं कि कौन सी लाइट है, कौन सा नैनोमीटर है जिसको हम डालते हैं और दैट गिव्स द मैक्सिमम एब्जॉर्प्शन या मैक्सिमम ट्रांसमिटेंस। तो इसमें भी कुछ वैसा ही है कि हम पर्टिकुलर, जैसे राइस के केस में या वीट के केस में या मेज के केस के अंदर पर्टिकुलर लेंथ का वेवलेंथ ही हम उसको डालेंगे या उसी वेवलेंथ की लाइट को हम कैप्चर करेंगे सो दैट वी कैन गेट द मैक्सिमम रिफ्लेक्टेंस ऑफ ए पर्टिकुलर क्रॉप सर, कि हम हमेशा एक रेंज मतलब थ्रो करते हैं।
बिल्कुल सही है सर, बिल्कुल सही है सर। ये आप जो बोल रहे हैं ये इसमें क्या है, जैसे अपन एंजाइमेटिक एनालिसिस में अपन आइडेंटिफाई करते हैं मैक्सिमम एब्जॉर्बेंस या मैक्सिमम ट्रांसमिटेंस अपन देखते हैं, तो उस ऑन दैट बेसिस अपन वो वेवलेंथ को भी कैरेक्टराइज करते हैं और कैलिब्रेट करते हैं इंस्ट्रूमेंट को। तो सेम उसी के बेसिस पे यहाँ पे भी हम स्पेक्ट्रल सिग्नेचर्स आइडेंटिफाई करते हैं कोई भी क्रॉप का, कि कौन सा लाइट जो है ये मैक्सिमम रिफ्लेक्ट कर रहा है और किस वेवलेंथ की लाइट को ये मैक्सिमम रिफ्लेक्टेंस आ रहा है। जैसे मैक्सिमम रिफ्लेक्टेंस हो सकता है रेड में भी आए, इंफ्रारेड में भी आए, वो हम आइडेंटिफाई करते हैं और पीक्स आइडेंटिफाई करते हैं। इसमें कौन कौन सी लाइट के पीक्स हैं, तो वो पीक्स जो होती है दैट इज वेरी स्पेसिफिक टू द पर्टिकुलर क्रॉप। और अगर हमने वो पीक आइडेंटिफाई कर लिया कोई भी क्रॉप का, तो we can design a sensor, we can upload the sensor, माने उसके बेसिस पे हम that particular sensor only we can upload in our instrument, we can change, because sensors जो होते हैं इसके sensors अलग से आते हैं, तो वो हम sensors लगा सकते हैं इसमें। इंफ्रारेड का सेंसर, आरजीबी का सेंसर, विजुअल सेंसर, इस प्रकार के सेंसर्स हम उसमें लगा सकते हैं। ये देखना पड़ेगा अपन को कि कौन सी लाइट वो सबसे ज्यादा रिफ्लेक्ट कर रहा है और पीक्स कौन कौन सी लाइट में आ रही है।
ओके, थैंक यू सर, थैंक यू सर।
हेलो।
हाँ सर।
हाँ, this is Rajendra Singh Thakur. Just I want to query some. There is a slide you have given that protein content can also be detected through remote sensing and GIS. So I am quite curious about that. because of subject in science and technology. So how that will implicate through GIS?
सर इसी वाले क्वेश्चन को मैं जरा और भी पूछना चाह रहा था सर कि इतनी ऊपर से सर एक छोटे से ग्रेन का प्रोटीन कंटेंट मेजर हो सकता है?
प्रोटीन कंटेंट कैसे करना है?
सर इतने सर देयर आर सम डिफरेंट प्लेटफॉर्म्स, अलग अलग प्लेटफॉर्म है सर। जैसे लैब के लिए अलग प्लेटफॉर्म है। लैब में आपको जो है डिस्टेंस जो है अपन को बहुत ऊपर नहीं रखना, बहुत पास रख के करना पड़ेगा। प्लेटफॉर्म्स अलग अलग होते हैं। फील्ड के लिए अलग प्लेटफॉर्म है, लैब के लिए अलग प्लेटफॉर्म है। तो ये जो मैंने आपको हाइपरस्पेक्ट्रल सेंसर्स को जो यूज करते हुए जो प्रोटीन का आइडेंटिफाई किया था, वो फॉर दैट दे हैड यूज्ड द लैब प्लेटफॉर्म, माने स्टेबल प्लेटफॉर्म था वो एक जगह खड़ा कर दो और उसके ऊपर नीचे ग्रेन रख दो और उसके ऊपर से आप अगर मान लो लाइट को, एक्टिव लाइट हो या पैसिव लाइट हो, वो देके अपन उस ग्रेन के प्रोटीन कंटेंट को आइडेंटिफाई कर सकते हैं। वो लैब के लिए प्लेटफॉर्म ही अलग है उसका।
No, I studied a paper few days back. I studied a paper that they had studied the protein content before harvest. So I think that GIS is remote sensing is also can be used through this. So if you have any,
उसमें ऐसा है कि जो लैब के लिए जो है, लैब में तो उन्होंने सिंगल कर्नल में निकाला है, सिंगल कर्नल्स को लेके निकाला है। और जो आप बोल रहे हैं प्रोटीन कंटेंट, वो जो है वो लीफ में निकाला है, उनने लीफ में आइडेंटिफाई किया है। फील्ड में अगर क्रॉप में ग्रेन में तो आइडेंटिफाई कैसे करेंगे? ग्रेन तो कवर रहता है तुम्हारा पूरा,
वही तो
उसमें तो ग्रेन का तो आइडेंटिफिकेशन हो नहीं पाएगा ना।
चलिए मैं उसको एक बार पेपर डाउनलोड करके पढ़ूंगा, देन वी विल हैव सम डिस्कशन अलोन।
बिल्कुल बिल्कुल डॉक्टर साहब।
ओके ओके, थैंक यू।
ओके ओके। Anybody else want to discuss?
शिवरामा।
हाँ मैडम, नमस्ते मैडम।
हाँ
नमस्ते मैडम।
नमस्ते। हम ये पूछ रहे थे कि ओनली प्रोटीन का इसमें निकाल सकते हैं या कोई और भी न्यूट्रिएंट्स हैं जो वी कैन कैलकुलेट, मिनरल्स और अदर टाइप ऑफ मेटल्स, मिनरल्स, अदर प्रॉक्सिमिटी प्रिंसिपल्स, ओनली प्रोटीन?
अभी तक तो मैडम प्रोटीन निकाला है और शुगर निकाला है इसमें, जहां जो मैंने लिटरेचर सर्वे किया है तो प्रोटीन और शुगर का तो मिला है। मिनरल्स का और सोडियम और जैसे आप जिंक और ये जो आयरन बता रहे हैं, तो इसका अभी तक कोई लिटरेचर मिला नहीं है इसका।
अच्छा, एक क्वेश्चन और था मेरा कि इसमें सिर्फ आइडेंटिफाई होगा या क्वांटिफाई भी होगा?
नहीं, क्वांटिफाई भी होगा मैम, क्वांटिफाई भी हो जाएगा। वो आपको ग्राउंड ट्रूथ माने आपको लैब में एस्टिमेट करके और उसके बेसिस पे आपको कैलिब्रेट करना पड़ेगा इस टेक्नोलॉजी को भी। जैसे हम फील्ड में हम स्पैड को भी करते हैं तो हम कैलिब्रेट करते हैं, फिर उसके बाद हम उसको यूज करते हैं। उसी प्रकार से कैलिब्रेट करने के बाद ही मतलब कि हम इसको यूज कर सकते हैं।
जैसे
जब एक बार कैलिब्रेट हो जाएगा, इक्वेशन डेवलप हो जाएगी, तो उसके बेसिस पे फिर हम यूज कर सकते हैं इसको।
ओके, ठीक।
सर अभी हमारे पास कोई इस तरह का इमेज कैप्चरिंग डिवाइस है क्या सर, हमारे मतलब हमारी यूनिवर्सिटी में सर, नाइब के अंदर?
सर अभी एवरीथिंग इज एट द एडवांस्ड प्रोक्यूरमेंट स्टेज है। all the instruments we had done the tender and everything is at the advance, मतलब within one month or 2 months each all the instrument will come। फिर उसके बाद उसको we will standardize the instrument in our lab or in the field and then we will be in a situation to use it। अभी तो कोई भी इंस्ट्रूमेंट जो है आया नहीं है इसमें, क्योंकि उसमें बहुत सारी हर्डल्स थे, इस वजह से थोड़ा लेट हो गया।
ओके सर, ओके सर।
Anybody else?
सर ये प्री असेसमेंट टेस्ट लिंक मैंने ग्रुप में शेयर किया हुआ है, so all the participants please attend the test।
अरे सौरभ साहब, एक वो था उसमें कि मेरे ख्याल से ना उसको व्हाट्सएप ग्रुप में डाल देते हैं, क्योंकि मैंने क्लिक किया ना तो वो एक्सटर्नल वेब साइट का वो करके दिखा रहा था। तो मेरी रिक्वेस्ट है कि उसको व्हाट्सएप पे डाल दीजिए तो वो ओपन हो जाएगा इजीली।
ओके सर, मैं व्हाट्सएप में अभी शेयर
हाँ, ठीक ठीक।
So as the discussion part is over, so I like to thank PI of the project, Dr. R.K. Nema sir, and the program coordinator, Dr. Shailesh Sharma sir, Dean College of Agriculture Engineering, for giving me an opportunity to interact with all my fellow friends and the seniors in this through this lecture. And in future also, if anybody wants to collaborate and work in this aspect, they feel free to contact me and the Nema sir also, because our group mandate is that we want to develop the human resource in the field of the remote sensing technology. Thank you.
Thank you, Shivram sir. It was very nice and comprehensive presentation. And to all the participants, we have also a session at starting from 3 to 5 p.m., 5:30 p.m. So we will play a learning video on the same period. So please be attentive at 3 p.m. Please join the same link. Assessment test. Assessment test कैसा कह दिया? जी. So thank you very much. Please do the pre-assessment test, and please be there at 3 p.m.